Our next speaker is Dr. Isobel Webb from the University of Bristol, who's going to talk about replacing SARS-CoV-2 ORF3a gene with fluorescent reporter genes does not produce faithful reporter viruses.
Hi everyone, so I'm going to be talking about some work in our lab in which we've replaced ORF3a with either M scarlet or M neon green, as you can see here. So highlighted in the yellow is the important images in which you can see the M neon green virus produced a beautiful fluorescent signal. But interestingly, when we used M scarlet, we didn't pick up any red fluorescence in either vero cells or in AATs. We included an ORF6 replaced with M scarlet virus as a control, in which you can see that the reporter is clearly visible. To then check whether the M scarlet protein was being produced at all, we counterstained with an M scarlet antibody, which is the panel beneath. And you can see that in vero cells, which is the top panel, you can pick up some M scarlet signal, but you can't in AATs, indicating that there is a non-fluorescent M scarlet protein being produced in these viruses. So we then looked at the genetic stability of these viruses, and we found that the M neon green was stable after 5 passages in both A549 and vero cells. But interestingly, we couldn't pick up the M scarlet virus at passage 5 in A549s. And in VTNs, in 2 out of 3 of our replicates, we got a large deletion of the M scarlet sequence. It's important to note that this large deletion included the chromophore, so this is potentially an explanation for the non-fluorescent M scarlet protein present in the cells. So we then wanted to investigate the replication kinetics of these viruses and found that in all of the cell types we tested, we found an attenuated replication phenotype for both of the recombinants we generated. Interestingly though, when we also included our additional deletion of the M scarlet sequence, we found a slight recovery of this attenuated phenotype, indicating that there's something to do with the M scarlet sequence that is also attenuating these viruses. And therefore, loss of ORF3a and presence of M scarlet is causing this attenuation. So I would like to thank Max, who I did this project with, and Andrew.
Oh, good.